Good morning, everyone. Good afternoon, everyone. So we have the pleasure today of having Dr. Lucia Tabaku. She will be our next presenter under the REYES. So Dr. Tabaku is Associate Professor of Statistics in the Mathematics and Statistics Department at Old Dominion University. She obtained her PhD in 2014 from Pennsylvania State University. And Dr. Tabaku is working on data recorded by wearable technology. And she is collaborating with biostatisticians from wearable and implantable technology from different groups, in particular from Johns Hopkins University group. So Dr. Tabaku, the floor is yours.
Thank you, Noro. Let me share my screen. Okay, so as Noro said, I'm dealing with accelerometry data. So and especially accelerometry data that was used in NHANES. So the talk is divided in three parts. The first part is I talk about accelerometers in general. And in the second part of the talk, I'm introducing NHANES, which is a big national study. And the third part will be focused on some of the research ideas that my collaborators and I are working on. So accelerometry data and accelerometers are part of this wearable, what we call wearable technology. And I was curious when this concept started, when was proposed. So it was proposed in 1960 by this mathematics professor at MIT, Edward Thorpe. And together with his colleague, Claude Shannon, they developed these systems to predict roulette wheels. Okay, so here I have a picture of some of the wearable devices. So for example, you have Fitbits, you have glucose monitors, you have Apple Watches, you have Google Glasses, and many, many other devices, wearable devices. The data, so all these devices are smart devices and small that can be attached to the human body, and they record the human signal. So all this data, all this signal can be transmitted to some apps on your cell phone, and then they can be integrated into the patient health records. So the application of these wearable devices in healthcare can contribute to a new approach in medicine, which can be preventive, predictive, personalized, and participatory. This is something that in the literature is called as the 4P medical model. So these devices, these wearable devices, can have a significant role in advancing precision medicine and can facilitate the emergence of connected healthcare. Okay. So here I have another picture where with some types of wearables and what they can do, what type of data they can record. So for example, we can have some wearables that are in this form of headbands, and they record EEG signals. So with these, what these EEG signals can do, they can measure the level of mental attention of a person. Other types of wearables are these sociometric badges. They can measure actually the level of social interaction of a person via a Bluetooth or a Wi-Fi devices or a Wi-Fi device. Other wearables are the smartwatches, for example, that everybody might have one by now. With a smartwatch, what can we do? We can measure actually physical activity. We can measure the sleep pattern of a person. We can measure the heart rate of an individual and many, many other things. There are also wristbands that can measure the level of stress of a person. Any other wearables can be embedded in clothing. Yeah, so these are sensors that are embedded in clothing, and they can measure, for example, the muscle activity of a person. Okay, so here I have 4 major groups of applications of these wearable devices in medicine. So these devices can be used in health and safety monitoring. So here I have groups of people that require special attention, for example, or the older adults, children, pregnant women. So these devices, this group of people can wear these devices and gait, posture, walking speed, respiratory rate, blood oxygen, heart rate, and all these things can be monitored, measured to ensure a timely help and treatment. Other area of application in medicine can be chronic disease management. So for example, here you can have devices that measure the blood glucose of a person, continuous monitoring the blood glucose, or continuously monitoring the blood pressure of a person. Other area can be disease diagnosis and treatment. So there are these devices that can early detect Alzheimer's disease, or they can monitor obstructive sleep apnea. And the other area can be rehabilitation. So for example, we can have wearables that are like glasses for the blind people, a smart device, smart hearing aids, and intelligent prosthetics. So this is developing these wearable devices and collecting data and understanding the data recorded by these devices is a new area of research, and many things can be done in this field. Okay. Okay, so here, so I talked a little bit about these wearable devices and accelerometers. So accelerometers, what are they? They measure they measure acceleration relative to Earth gravitational field. So basically, these accelerometers have 3 axes: up, down, left, right, and backward, forward. And they measure movement. So we use them in all these experiments to measure the level of physical activity. So the next video has 35 seconds and will show you, it was recorded by Jacek Urbanek. He's an assistant professor at Johns Hopkins University. And he recorded his student, Marta Karas. She's doing a PhD in biostatistics, and she's doing a lot of research in this area of accelerometry data. So in this video, Marta is wearing a right accelerometer and a right ankle accelerometer and a right wrist accelerometer. And she's doing all kinds of movements. For example, she is walking, she is clapping, she's jumping, and at the end of the video, she's putting on her jacket. So you can see on the right hand panels, you have 2 panels, and you can see the recording data from the right ankle accelerometer and from the right wrist accelerometer. And you can see what type of data they record. And when, for example, when she's putting on the jacket, you can see that the accelerometer that she's wearing at the right wrist records more intensive data. Yeah. And the one that she's wearing at her right ankle is almost does not record any type of data. So now I'm going to play the video. So here is Marta, jumping, clamping, walking, and the 2 panels that I mentioned to you, the right wrist and the right ankle. So while she's walking, you can see some more activity in the right ankle accelerometer. Now when she's jumping, you can see in both panels data. And now she is putting her jacket, and almost no activity in the right ankle, but a lot of activity in the right wrist. Okay. So I have the next 3 slides. The next 3 slides are about more data, how we represent data, this data that comes from accelerometers, how we represent it using a computer, using statistics. Yeah, so we have these three slides that I have show some pictures that I borrowed from Marta's paper. She wrote this paper on accelerometry data in health research, challenges and opportunities, was published in 2019. So what she's showing in this graph here, you see there are 3 panels. The first panel is the data that comes from an accelerometer with 3 axes, and the accelerometer was located on the left wrist. So you have 3 colors which represent each axis of the accelerometry data, and the data here is recorded for 24 hours. So it's kind of messy. You cannot see exactly all those 3 colors, yeah, that correspond to 3-dimensional time series, that correspond to the 3 axes that I was talking about. But what you can see here is this red dashed line rectangle, which represents 1 hour of data. So then what in the middle, in the panel in the middle, you can see this hour here, this hour of data, represented it in 60 minutes. Yeah, so you have a better and clear picture of the data and the 3-axis, 3-dimensional time series of accelerometry data. And now you in the second panel here, you see this rectangle, yeah, which represent actually 1 minute. So you have 60 seconds here of data. So in the third panel here, you see that those 60 seconds that were recorded in the middle panel. So here, when you represent the data at the second level, you have a better picture, a better understanding of the data, and you can see all 3 colors that represent the 3 axes of the accelerometer. So the goal is to use this. The idea is to use this raw data at the second level to get a better understanding of it. Next picture shows. I have 2 subjects here, subject 1 and subject 2. They all wear an accelerometer located on the left wrist, and they perform 2 types of movements. One is walking and the other one is dressing up. So you can see that while they are walking, these 2 show like this periodic pattern, but there are some differences in this periodicity from one cycle to another, depending on the subject. Yeah. Now, when we look at the other type of activity, which is dressing up, we see a lot of randomness here. Yeah. So with this picture, I want to show you that there is a lot of variability between subjects. Subject 1 can walk differently than subject 2, and also a lot of variability within the data that comes from each subject. So there is a lot of variability in the way the subject 1 dresses up, and there is a lot of variability in the way the subject 2 dresses up. Yeah, so what I'm saying here is that movement, or for example, walking, can be different for the same person. If walking can be different if the person goes to the kitchen in the morning, or if the person runs to a meeting in the afternoon, yeah, runs to get in time to the meeting. So the walking can be different. Even if it's the same person, the walking can be different. Dressing up is the same, yeah, so it can be different for each person. It can be different from one person to another. So what I'm here, I want to show you that there is a lot of variability between subjects and between subject. So a lot of variability from one subject to another, but also a lot of variability for each subject. Next slide is what do I have here? I have, again, accelerometry data that comes from accelerometer located on the hip. So I have two locations of the accelerometer. I have a location on the hip and then a location on the left wrist. And there are 3 activities that are done here: dealing cards, dressing up, and walking. And the goal is to show you that the location of the sensor can be different, and and different based on the activities. So for example, if the person is dealing cards, the hip accelerometer cannot detect that movement, but the left wrist accelerometer can detect a lot of movement. With the next activity, which is dressing up, there is some detection of the activity in the hip sensor, the hip accelerometer, and a lot of activity in the left wrist. And then when it comes to walking, we see activity in both sensors, yeah, in both accelerometers. So if we want to use these type of accelerometry devices to predict movements, for example, we have to take into consideration the location of the sensors, different activities that are performed, the variability within subjects, the variability among subjects, and many, many other things. So the goal of this type of research is to understand how these, to understand this type of data. Yeah. Okay, so that was the first part where I talk about accelerometry data in general. Now I'm going to switch to the second part where I introduce NHANES. So what is NHANES? NHANES stands for the National Health and Nutrition Examination Survey. So if you Google this, you'll find a lot of information about this study. Many people use this study because the data is made public after a couple of years after it was recorded and cleaned up. So if you Google, you can see that this study, NHANES, is conducted by the National Center for Health Statistics in the United States. And the National Center for Health Statistics is part of CDC. So CDC stands for the Centers for Disease Control and Prevention. So if you open this web page, you can see a lot of information about the study. For example, how they survey their participants. They have a lot of documentation about the data, how they collect the data, and they also have the data itself. So you can download the data from their website. And then you can access some of the publications and the products that were based on on this data. And they also have some data analysis tutorials that they help you how to do the analysis. I think they have code in SAS, in R, and many other softwares, statistical softwares. So you have a lot of information, but it takes time to understand this information and to be able to use it. Another good resource is their search variables link. You can look. For example, I was searching for physical activity, and then I got 22 results. And here you can see that NHANES was recorded. Data for NHANES was recorded from up to 2020. Yeah, and here I have the NHANES 2003-2004, but if you scroll down, you will see data since 1960. Yeah. Okay. So I told you about NHANES, who conducts this study, who organizes. But what is the goal? Why people bother to collect data in NHANES? Why so much trouble? Yeah, because it's a national study, so it requires resources. So why do we bother in spending so much money and spending so much resource on this? So the goal of the study is to evaluate the health and nutritional status of the United States population. And if you Google and look on the CDC webpage, they say that the goal of this study is to help develop sound public health policy, direct and design health programs and services, and expand the health knowledge for the nation. Yeah, so collecting data every year on the national on the United States population, the government has an idea about the trends of the health and nutritional status of the people in the United States. Okay, so they collect data on people, yeah, people from United States. But how do they choose these people? Are we picking people from every state? Are we sampling people from every state randomly, or do we do this in a more organized way? So there is an organized way, and that way is called sampling design in 4 stages. So this picture shows you the 4 stages of sampling people, sampling the participants in this study. So it starts with stage number 1. So in stage number 1, statisticians at CDC, for example, gather together and they divide the map of the United States into strata. So these strata, they create this strata based on the geography and the proportions of minority populations. Yeah. So let's say they already have these strata. So now from each strata, they sample using, again, probability theory and statistics, they sample these counties. Yeah. So from each strata, they sample counties. These counties are called primary sampling unit, or short PSU. Yeah. So at stage number 1, they sample these counties or these PSUs. PSU, the primary sampling unit, is something that we use in sampling theory courses in statistics. Okay, so let's say that we already sampled the counties at the first stage. Now, what's the second stage? The second stage, at the second stage, they sample the segments from those from those selected counties at stage number 1. Yeah. What are these segments? These segments are actually cities. Yeah, cities within those counties that were sampled at stage number 1. Stage number 3, so out of those sampled segments, they sample households. Yeah, this is done at stage number 3. And out of these sampled households, they sample individuals in stage number 4. Yeah, so you see that at stage number 4, we already have our sampled participants, but they are not done randomly. So they have to follow this cycle here, this cycle in four stages. Okay. So when I told you that the territory of the United States is divided in strata by geography and proportions of minority populations, then what statisticians do, they sample usually 2 PSUs from each strata. Another characteristic of this enhanced study is that participants are under the age of 18 and participants are of over 60 years old are oversampled. Also, Mexican Americans, African Americans, and low-income white American groups are also oversampled. Why do we bother to do that? Well, we have a statistical reason behind this. Yeah, we want to increase the reliability and the precision of the estimators that we calculate from this data. Yeah. So everything is done with a reason. This sampling theory helps us to do the sampling, to select our participants in a statistical way. Okay. So far we have our participants in the study. But how do we collect the data in this for these participants? We have our participants, but how do we ask them questions? How do we measure different health outcome? How do we measure their health in general? Yeah? How do we quantify their health? So the data is collected through interviews and physical exams. So what are these interviews? These interviews can be done by phone or by meetings. And these interviews, participants are asked different questions about demographic, socioeconomic status, their diet, and all kinds of health-related questions. At the physical exam, they are actually they actually run tests on these people, medical tests, dental tests, physiological measurements, and lab tests. Are they willing to do this? Some people are willing to be participants in the study because they receive compensation, and the medical reports that they have are given to them. Yeah, so all these analysis, medical analysis are given. All these medical reports are given to the participants. How about the data that we collect? All this data on each person. This data is confidential, yeah, and is protected by public law. Okay. So once we have the participants, they are paid. We can tell them that their medical records are. We can give them the medical records and so on. But we have to get their consent. Yeah, we have to send them an invitation letter to participate in this enhanced study. And if they agree, then they go through all these interviews and physical exams. But then even if they accept to be part of these interviews and physical exams, they have to complete this online questionnaire and make sure that they are eligible to be part of this study. So as I said, the health interviews are done over phone. But how about the medical exams and the lab tests and so on? All these medical tests and lab exams are done through a visit to the mobile exam center. Yeah? So these mobile exam center have all kinds of how should I say, medical devices that can measure, for example, height, weight, blood pressure, glucose, cholesterol, exposure to environmental chemicals, and so on. So what are these mobile exam centers? So they look like this. Yeah, they are these trailers or vans or whatever you want to call them. So the team, the medical team and the technical team from CDC and people that are involved in organizing and collecting data in ENHANCE, they use these and they drive across the country, and they drive in those counties that were sampled, and they schedule visits of those people that were accepted to be part of this study. Yeah, so the physical exams take place in these mobile centers. So these mobile centers are equipped with advanced computers that collect and process data, and then the data is transmitted to CDC within 24 hours of collection. Okay, so now we know that we have how we get our participants in ENHANCE, how we collect our data, yeah, through interviews and physical exams. But what do we do with this data? Yeah. So what we do, we CDC and ENHANCE study make it public. They make it public. Yeah. But after a couple of years, it's not done immediately. It's not going to, you cannot access, for example, right now, you cannot access data from 2021 or from 2020. Yeah. You need a couple of years. But once it is made public, universities, researchers from all kinds of universities, from healthcare providers, from institutes, or from all kinds of research organizations can access the data, and they can use it to do the data analysis and answer all kinds of research questions. So what are these? Do we need this type of research? Yes, we need. And why do we need it? For example, here I found some examples where that contributed to some changes in the health in the United States. So, for example, based on this data that was collected in ENHANCE, growth charts were created, and these growth charts are used nationally by pediatricians. Based on the analysis of this data, it was developed policy that eliminated lead from gasoline and food and soft drink cans. Based on this data, because it's collected for such a long time, they can see trends in obesity and they can monitor these trends and they can increase the programs focused on diet and exercise. Based on this data and on the analysis of this data, they actually increase the public awareness on diabetes, for example. Based on this data, they increased prevention programs to reduce hypertension, cholesterol level, and the association with heart disease. I think heart disease, death from heart disease is the leading cause in US, maybe? I think so. I think heart disease and then cancer, or the other way, anyway. And another benefit of using and analyzing this data was the decision to fortify the food supply with folic acid to prevent birth defects. So this is useful, and analyzing this type of data has some useful consequences on the health care in the United States. Okay, so now when did everything start? I mean, how old is NHANES? For how many years was collected by now? Or when everything started actually in 1960. And it started in 1960 and was done periodically up to 1994. And then from 1999 was continuously collected. Yeah, data was continuously collected and NHANES was continuously organized. So I said that the first NHANES was organized in 1960, but it started with the National Health Survey Act that was passed in 1956. And here on this slide, I just have a few things on the improvements that they did from 1 year to another. So for example, in 1960 to 1970, and the focus that they had in those 10 years. So the focus here was on growth and development of children. If we move forward to 1988 to 1994, actually they, excuse me, they focused on Black Americans and Mexican Americans. They focus on children, small children. They focus on older adults, and they focused also on the emphasis on of the link between environment and healthcare. Yeah, so at that time they consider measurements on level of pesticide exposure, certain trace elements, and so on. What happens? Okay, so we've seen it start in 1960, but what happened with this pandemic? Actually, many things happened with this pandemic, and we will see the consequences of this once it passes and we want to analyze the data. Because of this pandemic, actually, they had to pause everything. They had to pause the data collection. And what does it mean? If they pause the data collection now, they have to do all kinds of changes in the sampling design. By the sampling design, I told you when you have those 4 stages. So now they have to change that sampling design. They have to change their questionnaires. They might need to add some other questions, for example, vaccination, or they have to add if people had already COVID and what happened to their health. Yeah? So all kinds of questions related to the coronavirus pandemic. And also they have to change the exams and the lab procedures. Yeah. So, and because there were so many changes because of this coronavirus, actually CDC and the National Health Statistics had to write and report all the changes. So here is the publication from the National Health Statistics that was published in 2021, and they explain everything on this data and this pause due to coronavirus pandemic. So here in this paper, they explain what are the changes and how we should work with this type of data, the new data that was collected during the pandemic. So if we want to work with this type of data, we have to read all this material and make sure that we are doing the right thing. Okay, so I talked about the first part. I talked about accelerometry data in general. In the second part, I talked about enhance. Now I want to bring in the accelerometry data in enhance. So do we have accelerometry data in enhance? Because so far I told you that enhance collects the data on health, yeah, on all kinds of questions related to the health of participants. Well, enhance collects data from accelerometers. So there are and accelerometry data was collected in 2003, 2004, and 2005, 2006. Yeah, so NHACE was the first study that made public this large data set of physical activity. So the way they collected this accelerometry data was by this hip-worn actigraph accelerometers. The participants in enhance had to wear this hip-worn actigraph for 7 days, 7 consecutive days, during the waking hours, with the exception of swimming and bathing. So that was done in 2003, 2006. Yeah, they used these actigraph accelerometers. Well, later on in 2011, 2014, they introduced wrist physical activity monitors. Yeah? So the data was collected in 2011 up to 2014, but it was made public in 2020. So you see, it takes a couple of years until we get access to the accelerometry data. Another another study that is worthwhile mentioning is the UK Biobank. This is another public study. It's a large one, but is collected on UK participants, people from United Kingdom, and the data is it's public, but not like enhance. If you want to use it, you have to ask permission. Yeah, you have to register, and I don't think you have to pay, but I'm not sure. You may have to pay some. So enhance is free. Yeah, you can access, you can download the data, and you can use it the way you want. You don't have to ask any permission. Okay, so I said 2003 was the first time when in enhance they introduced accelerometry data, but they did not, and they collected physical activity data based on these accelerometers. But what happened before these accelerometers? Before these accelerometers, what they did was to ask questions about physical activity. So they asked these questions through questionnaires. But there are some problems when you use questionnaires to find out about the physical activity of participants. It has limitations. So, for example, people cannot remember how much they exercised, yeah, when they exercised, or they cannot quantify the intensity of their physical activity. When it comes to children, the data on the physical activity for children has to be provided by their parents. Yeah, parents may not remember when their kids exercised. Yeah, so the data for kids can be incomplete, and children can play outside, can play away from their home. So again, parents are not aware of that type of physical activity. Yeah? So using questionnaires for collecting data on the physical activity has some limitations. So what I'm trying to say is that collecting data on physical activity using accelerometers is much better. Yeah, it can be done continuously because they wear the accelerometers for a couple of days, and it is objective. Yeah. Okay. So this ends somehow the second part of the talk that I mentioned to you about enhance and accelerometry data in enhance. And now I'm going to focus on a couple of ideas that my collaborators and I are trying to answer, yeah, and try to do research on these ideas. So what do we care about? We care about physical activity and mortality. Yeah. Why do we care about that? Because physical activity is a major determinant of human health. There are many studies published in this area, and all these studies say that insufficient physical activity is a risk factor for chronic diseases. You don't exercise enough, then you end up with some cardiovascular diseases, diabetes, cancer, and so on. Many studies that were published show that physical activity is a predictor of mortality risk, as age, gender, ethnicity, and many, many other variables. So why do we care of investigating this physical activity? Yeah? Why do we care to understand if the volume of physical activity measures, the time we do the physical activity matters? Why do we care the intensity, the level, the timing? Why do we care about all these things? Because physical activity is modifiable. Yeah? So we cannot change our age, but we can change the physical activity that we do. Yeah? We can modify the level, the volume, the time, and that contributes to our well-being in general. So the type of questions that we ask is the type of questions that we ask are, for example, what is the association between physical activity and health outcomes and mortality? Can we predict mortality based on different covariates and physical activity covariates? Can we rank the most predictive covariates? Can we build prediction models, simple prediction models that can be used, for example, in a doctor's office? So these are the type of questions that we want to answer in our research. Okay, so while trying to answer these questions, we realized that there are some challenges when it comes to working with NHANES. And these challenges are challenges with data. So why do we have data? No, it's made public. So why there are challenges there? Because nobody tells you actually how to work with this data. Yeah? And it takes a non-trivial amount of background information. So if you want to download the data, you have to make sure that you understand what that data is. Yeah, what are the questions that were asked in NHANES study, and then you want to work with accelerometry data, you want to work with mortality data, you want to work with data on their health, like you want to see if they have diabetes, if they have cancer, if they have, so all these variables have to be downloaded from the NHANES website, and then requires a lot of data processing, cleaning, and linking. You have to link all these files. Yeah. So this requires some effort. Then what are the other problems? What are the other challenges? There are some challenges with the complex survey design. So I told you that the data, the participants are sampled in 4 stages. Well, with every stage, we create some sampling weights. Yeah? So for example, for each participant that is part of the NHANES, we'll have attached or assigned a sampling weight. So what is this sampling weight? It's actually a number that is computed based on the probability of sampling, yeah, at every stage. So this sampling weight is a measure of the number of people in the population that are represented by this participant in NHANES. So do we care about these sampling weights? Yes, we do care a lot, and they should be incorporated in the prediction models that we create. They should be accounted for in the modeling part of the analysis. Why do we care about these sampling weights? Because incorporating them in their statistical analysis, we obtain better estimates. Yeah. And another challenge is with the accelerometry observations. So in NHANES, for example, I told you that NHANES 2003-2006, they collected data on accelerometry data. They collected accelerometry data, and the participants had to wear these hip-worn actigraphs for 7 days, for 7 consecutive days. And the data was recorded for 7 consecutive days for 1,440 minutes, so basically 24 hours. So now we have this data on each participant for 7 days and for 1,440 minutes. Yeah. So what do we do with that data? How do we organize that data? So we have to find smart ways to summarize that data, and in the same time to capture the high dimensionality information that we have in that data. And there are also some quality issues, quality of the data, that we need to take into account. And there are algorithms that help us to get the best data, from these accelerometry observations. So what I have here are is the data that you can actually download from NHANES. Yeah, so data from the accelerometry data from the NHANES website. So you can see that each participant in this data is identified through this sequential number. So 3112 is 28 is one participant, and for each participant we have activity counts. So the accelerometry data is measured by these activity counts. So we have every day, day number 1, and then every minute from minute 1 to minute 1,440, and then we have activity counts at every minute. So it has this long format that cannot be used, for statistical analysis. So we have to reduce the file. So my collaborator Andrew Leroux, he's an assistant professor in Colorado now. He was able to transform the data and now reduce the entire file by 80%. So how did he reduce the data? So here we have the first participant. Let's say we have the 7 days of data, and we have minute 1 up to minute 1,440. So for day number 1, we have all those minutes and all those activity counts at every minute. So basically at minute number 1, which is I think midnight, this participant had 166 activity counts. So that is 166 is what the accelerometer recorded. At minute 2, the accelerometer recorded 27 counts. Of course, there are some issues with the data. We have missing data. We have quality flags. We had problems with the calibration of these devices and so on. So I'm not going to focus on that, but there are some publications that help you to answer those type of questions. So NHANES 2003-2006 collects activity data. So here I sampled 2 subjects. One is red and one is blue. And I plotted their activity counts, their row activity counts. So we see the blue subject has more activity in the first part of the day and then before 4 p.m. And then the participant in red had more activity in the second part of the day. This is the activity counts for these 2 participants, day 1. Day 1 means Sunday. They started the experiment on Sunday. Then having all these, you see these activity counts are somewhere between 0 and 5,000 for these 2 participants. So we wanted to have we wanted to take the log of this activity counts and reduce here from the range of the data from 0 to 8. But again, there is a lot of noise and we cannot make sense of this data. So what we did, we tried to remove the noise using some smoothing techniques. So using the smoothing techniques, we were able to obtain these nice and clean curves, which correspond to the activity profile of each subject for day number 1. So you see the high activity for participant in blue, and then the high activity in the second part of the day for the participant in red. Okay, so that was for Sunday, but remember in NHANES we have data for 7 days. So here I plotted all 7 days, all the profiles. for 7 days for the 2 participants. So you see there is a lot of variability from one subject to another, and there is a variability in the week, during the week. Yeah? So one curve here represents 1 day of the week, and we should have 7 curves for everyone. We have so Andrew developed to make life easier, yeah, and to improve reproducibility of the analysis. Andrew developed this R package called R-enhanced data. And actually you can, if you download this package, then you can have access to it. This package actually downloads the data from the CDC website, and it's clean data, and you can process and perform your analysis much easier. Yeah? Just a quick, I'm not going to follow the whole slide here, but I just want to tell you that we, in order to do the statistical analysis and to answer our research questions, we had to apply some exclusion criteria. Because we study mortality, we focused on those people between 50 and 85 years old. We included some predictors that are related to the physical activity. We included some predictors related to cardiovascular status, mobility status, diabetes, the history of alcohol consumption. So out of the 14,631 participants, this is the number of participants in Enhanced 2003-2006. After we applied all the exclusion criteria, we cleaned the data and so on, we ended up with almost 3,000 participants. So we did the analysis on these 3,000 participants. And here we have, so I said we are interested in prediction models. We want to see what are those variables that are the best predictors of mortality. Yeah, so what we did here, we applied some algorithms. These algorithms this algorithm is called the forward selection. And the stopping criteria is this red dot that corresponds to AUC, area under the curve. So all these variables that were selected here before the red dot are the most important predictors of mortality. Yeah, so you see that there are those in red are variables that were created using physical activity collected by accelerometers. Yeah, so age, the total activity count here, TAC is included. So there are education, diabetes, they are all important predictors of mortality. And then here is another plot that we created, is called the correlation plot, and shows us how correlated is the variable age with all the other accelerometry derived measures. And this plot was done by my collaborator Katya and published in 2019. So I will stop here because I think I'm, yeah. Any questions? Thank you.
Thank you for the talk. Yeah. Thank you, Dr. Tabaku. If you have questions, you can type them, or you can say those. Anyone has any questions? One thing that popped up in mind when you were speaking, can you describe more about the 3 orthogonal axes on the accelerometry?
That's a good question, Noru. Marta is doing a lot of research on that. What I know is that, so she's really into that type of research, because that type of research allows her to answer all kinds of questions, you know, predict movements, predict activities. So I know that it records three-dimensional time series of data. And those 3 axes are orthogonal axes that correspond to up, down, left, right, and then backward and forward. Yeah. But there are lots of papers in that area, right, dealing with raw data.
Thank you. Anyone else? Anyone has a question? Let me see the chat. Oh, one question also someone is asking is, how do you relate enhanced data with personalized medicine?
How do you
How relate enhanced data and what do you think of personalized medicine, if I reframe it?
actually in personalized precision medicine, yeah, is about personalized medicine. I think you need more genetic level, for example, data at the genetic level to do precision medicine. And I'm not sure if NHANES has that type of data. Genetic test, I don't know if they have. But it's worth searching for those variables.
Anyone else have any questions? So for the enhanced data on COVID-19, we have to wait for the next 2 years.
Actually, I don't know. I have to read it. I have to read the report that they published from the CDC and see what type of data they will make available to the public.
Okay. But interesting.
Only the accelerometer data, for example, the one in 2003, the one in 2011, 2014, when they used the wrist accelerometers, was published in 2020. And I know that my collaborators from Johns Hopkins are trying to get publications, you know, to be the first that are publishing in this area with the new data.
Yeah, very exciting.
So there, I guess there is a lot of competition among these research groups to publish papers on the new data.
And I want to remind, you know, our participants in REYES that there's some kind of mentoring program. You know, please sign up. These are very exciting projects, and you know, we always look for ways to solve the problems. You probably have very good ideas. You know, pair up with a mentor and try to see how you can solve any of those open questions. Again, very good rewards can come from it.
Yeah, if you are interested in health care and health-related questions,
right.
The work that I'm doing now is more on the biostatistical side. So we are interested in these variables related to health. And NHANES is public, so everybody can use it.
Anyone else have any questions? So it's almost time. So I want to take this opportunity to thank Dr. Lucia Tabaku again for coming and joining us today. I want to thank the organizers too. And I look forward to meeting the rest of the group in other meetings, in other talks.
Thank you, Noro. Thanks.
Anything else, we'll just stop it. All right, thank you all. Thank you, everybody.
Thanks.
Bye-bye.
Bye.
Bye.